Hello and welcome to Nikolay's genetics lessons, and in this video I'm going to talk about what is gene complementation group. In simple Mendelian genetics, as you remember, most of the examples are about one gene and one trait, and one gene can be dominant or can be recessive. We call such homologous genes alleles. So two alleles would be just different forms of the same gene. So for example, this can be a gene that is dominant, capital A, and can be recessive allele, small a. Usually in simple Mendelian genetics, dominant allele code for the normal protein, and recessive allele would code for the defective allele. So in simple Mendelian genetics, recessive allele usually just mutant gene. So as you see, two alleles in diploid organism can make three different genotypes. The first one would be homozygous dominant, another one would be heterozygous, and the third one would be homozygous recessive. Let's now use example where dominant allele code for the red pigment and recessive allele produce defective pigment. So basically pigment wouldn't be produced, so the color of this plant would be white. So flowers would be white. That means that these two genotypes would produce plants with red flowers, and genotype that is homozygous recessive would produce plant with white flowers. So far it is very easy and simple to understand. What would happen if we would have one plant that is going to be heterozygous, phenotypically it's going to be red, and if we cross it with another heterozygous plant. So we have one plant that is heterozygous, we cross with another plant whose genotype is also heterozygous, and phenotypically both these parents produce red flowers, and the progeny phenotypic ratios would be as follows. So capital A here, capital A small a here, capital A small a here, and small a here. So as you see, phenotypic ratios in the following generation would be 3 to 1. So one genotype that is homozygous recessive would produce plants with white flowers. What would happen if we would use homozygous recessive genotype and would cross with another homozygous recessive genotype? 100% of the progeny would produce plants with white flowers. So you wouldn't be able to see any variation in flower colors here, because both parents genotype is two defective alleles that do not produce red pigment. So we would have 100% of the progeny that would produce plants with white flowers. But this is example when we have only one gene. If the trait would be under the control of more than one gene, we may have such situation when we have one strain of plants with white flowers, we cross with another strain also with white flowers, and such a cross would produce plants with 100% all red flowers. With this model, this is absolutely impossible to explain what we see here with such a model that we see here. And now let me show how this is possible. Imagine that strain number 1 and let this be strain number 2, have this trait under control of 2 genes. So let me show you one set of chromosomes here, and this is going to be second set of chromosomes here, and one set of chromosomes here, and second set of chromosomes here. So let's imagine that in the simplified model we have only 2 chromosomes for each flower. Each chromosome would have another homologous chromosome, so total number of chromosomes would be 4, but 2 would be unique and another 2 would be homologous chromosomes. So let it be chromosome number 1, and this is going to be chromosome number 2. So this is just different strains of the same species, and let red color would be red pigment would be under control of 2 genes. So in order for the red pigment to be produced, we need at least one normal allele for the chromosome number 1 and at least one normal allele for the chromosome number 2. But imagine that on the chromosome number 2 we have 2 recessive alleles. So red pigment wouldn't be produced, because if even this gene would produce normal protein, but by itself this protein wouldn't produce red pigment. In order for this protein to produce red pigment, another enzyme have to be produced and change its conformation, so it would become red pigment. But if second set of alleles would be mutated, so small a and small a here, so mutated here means that this enzyme wouldn't work properly and red pigment also wouldn't be produced. And in another strain we may have different situation when one set of genes or the same gene with 2 alleles would be defective. So would be, for example, small b, and by the way here we would have capital B and capital B, but another set of genes, another set of alleles that is going to be on the other set of homologous chromosomes would be normal. So capital A and capital A. Those normal enzyme would be produced, but normal protein wouldn't be produced. So it can be misshaped and our enzyme wouldn't be able to change its conformation in order to produce normal red pigment. So in both strains, as you see, this trait would be under the control of 2 genes, gene B and gene A, but if one set of alleles would be mutated, would be homozygous recessive here and homozygous recessive here, red pigment wouldn't be produced. But let's see what would happen if we would cross strain 1 with strain number 2. No matter which chromosome this or this parent would give to the progeny, both of them has normal B allele. So let's say that progeny would get this chromosome from the parent 1, and that chromosome has normal allele that produce normal protein, and from the second parent, once again, any of these chromosomes can be inherited, but both of them has defective allele B. So now, as you see, we have one normal allele from the first parent and one defective allele from the second parent. So here we have a heterozygous state for the gene B. And what would happen if progeny would get any of these alleles for the gene A? Once again, no matter which chromosome progeny would inherit, they would inherit from the parent 1 defective chromosome with defective allele small a that wouldn't produce normal enzyme. But no matter which chromosome their progeny would inherit from the parent 2, they would have a normal dominant allele A that would produce a normal enzyme. So as you see, 100% of the progeny, if we cross these two strains, would be heterozygous for the gene A and heterozygous for the gene B. So this chromosome with normal allele B would produce normal protein, and another normal enzyme A dominant would change conformation of this protein, and we would see red pigment. So as you see, 100% of the progeny would be heterozygous, and 100% of the progeny of these two strains would be plants with red flowers. So two parents that were with white flowers would produce 100% of the progeny with red flowers. And this is going to be what we would see in F1 generation. If you want to know what would happen in F2 generation, we have to self-cross or self-pollinate if it is plant, and genotype would be capital A, small a, capital B, small b in 100% of the F1 generation, and we have to cross with another same genotype, that is capital A, small a, and capital B, small b. Let this be, for example, parent 1 and this is parent 2. As you see, genotypes are the same, so both these parents would produce the same type of gametes. And what variants of gametes we may have here? We may have capital A and capital B here. So if parents diploid, their gametes would be haploid. If parents would have two alleles for each gene, gametes would have only one allele for each gene. So the second variant would be capital A and small b, and the third variant would be small a and capital B, small a, capital B, and the last variant would be small a and small b. So I used genotype of parent 1, but as you see, parent 2 genotype is the same. So parent 2 would produce the same four variants of the gametes. And as you remember, gametes are sex cells, that is haploid. In humans, we have two variants of the gametes. This is sperm and egg cell. So once again, if you remember, this is parents from F1 generation. And when we cross these two parents from F1 generation, we are going to get F2 generation. So let me clear space here. And now let's make a Punnett square with these genotypes. So this is going to be capital A, capital B here, capital A and small b here, small a and capital B here, and small a, small b here. And let's say this is going to be parent 1 and parent 2 genotype, or gametes that parent 2 genotype would produce would be the same. So on the side we put capital A and capital B here, capital A and small b here, small a and capital B here, and small a, small b here. Now, when we build a Punnett square, we would be able to predict all the genotypes in F2 generation. So this is going to be 4 by 4, and we would have 16 cells here with different genotypes, but let's see how many phenotypes we would have here. So in the first cell, this parent would give to the progeny dominant allele A, this parent also would give dominant allele A, this parent would give dominant allele B, and this parent also would give dominant allele B. So capital A, capital A, capital B genotype here, capital A and capital A here, capital B and small b here, capital A, small a here, and capital B, capital B, capital A, small a here, capital B and small b here, capital A here, capital B, small b here, capital A here, and small b here, capital A, small a here, and capital B, small b here, capital A and small a here, and small b here. And here we have capital A, small a, capital B, capital B, capital A, small a here, and capital B, small b, small a here, capital B, small a here, and capital B, small b here. And the last column, capital A, small a here, capital B, small b, capital A, small a here, and small b, small b, small a here, and capital B, small b, and small a here, and small b. So, in order for the red pigment to be produced, in each allelic pair, we have to have at least one dominant allele. So, this genotype here would produce plants with red flowers, plants with red flowers here. Those one allele is recessive, but we have another normal allele that would produce normal protein, and two normal alleles for the enzymes here. So next we also would have red normal phenotype here. This genotype also would produce red flowers, but next genotype, as you see, one of the genes would be in homozygous recessive state, and those we see here normal enzyme protein, normal protein wouldn't be produced, so normal pigment also wouldn't be produced. So this genotype would produce white flowers, and this genotype would produce red flowers, white flowers here, red flowers here, and red flowers here, white flowers here, and white flowers here, red flowers here, and white flowers here, and this genotype also would produce white flowers, and of course this genotype also would produce white flowers. So let's count what the ratio we have here. We have 1, 2, 3, 4, 5, 6, 7, 8, 9. We have ratio 9 to 1, 2, 3, 4, 5, 6, 7. So the ratio red to white flower. So red to white flowers that we are going to see in the F2 generation going to be 9 to 7. So what you see in this picture we call gene group complementation. Once again, when we cross one strain with another strain, and if trait is under control more than one set of genes, and if the first strain would have a homozygous recessive condition for one set of alleles, another strain would have a homozygous condition for another set of alleles, but in F1 generation these two strains would produce normal phenotype, because we see here gene complementation group. And if gene complementation group consists of two genes in the F2 generation, we would see this ratio. If this is going to be more than two genes, for example 3, 4, and we may see in F2 generation different picture. So this is one of the ways how to find how many genes control the trait. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.